Hi, I'm Aayushi. I'm a third-year BMed student.
I'm Maddie Jackson, and I'm a fourth-year BMed student. For our 2019 CERT project, we worked with Dr. Heylman to optimize and develop protocols for the loading, staining, imaging, and analysis of cells within a microfluidic device.
Microfluidic deals with the movements of very tiny amounts of liquids in controlled environments to help analyze biological and chemical processes. It helps with applications such as drug delivery and stem cell analysis.
Some of the methods we use include loading the microfluidic devices using two different methods, active loading and passive loading. Now we're going to demonstrate the different loading methods on a device using colored water. In an actual experiment, we would be using a cell solution in the sterile hood.
Once we loaded the cells, they were placed in the incubator for 24 hours to grow, and then we fixed the cells and stained them with Hooks, a fluorescent dye.
Passive loading of the devices involves placing an empty 200 microliter pipette tip into the outlet of the device and loading 200 microliters of cell solution, or in this case, colored water, into the pipette tip that will be inserted into the inlet of the device. Without depressing the plunger at all, I eject the tip filled with solution into the device, leaving it behind to flow on its own. The pressure head height formed by the media solution will cause the solution to flow into the device through the chamber until both tips are at an equal media head height. Active loading of the devices has a similar setup. An empty 200 microliter pipette tip is placed in the outlet. However, only 20 microliters of solution is loaded into the inlet pipette tip for active loading. The tip is inserted into the inlet of the device, and I slowly eject solution into the device until both tips have an equal height of solution. Without moving my thumb on the plunger, I eject the tip into the device. After staining, we use the upright fluorescent microscope to image the cells. In order to get a better view of the bottom of the device where the cells are adhered, we developed what we call the sandwich method. This method places the device on a microscope slide, which can then be flipped over and placed on a microscope stage. The stage was then moved manually to collect a series of images that were stitched together in Fiji to get a full view of the inlet, outlet, and chamber of the device.
Through this process, we learned how to optimize the existing protocol, develop a new imaging protocol, and begin characterizing the newest design for the microfluidic device.